for seminar provides a forum for presenting and discussing state-of-the-art research findings in HIV pathogenesis, virology, care, and treatment, and to provide an opportunity for communication and collaboration among HIV/AIDS researchers across the 5 CFAR affiliated institutions and their partner organizations. It's my pleasure to introduce today's speakers. We have 2 speakers. First up is our new faces speaker, Dr. Katrina Ortblad. Dr. Ortblad did her MPH at the University of Washington, followed by doctoral work at Harvard, and she returned to the West Coast for a postdoc at UW in global health. She's currently an assistant professor here at Hutch. Her research is in implementation science, and it aims to improve equity in healthcare access across models, in healthcare access through models of HIV service delivery in high prevalence settings. Her ongoing projects are based in Kenya, and they include a randomized trial on HIV self-testing, a study on pharmacy-based PrEP delivery, and a study aimed at increasing PrEP uptake and continuation among young women. Her research is multidisciplinary and spans epi, economics, public policy, psychology, and implementation science. And today she'll be talking about leveraging the private sector to expand the reach of HIV PrEP services in Kenya. So thank you so much.
Great, thank you, and thanks so much for having me. I had an introduction plan, but I can just skip now. So, but we'll get started. I was also going to introduce my research program. As was just mentioned, our research team focuses on research that aims to improve equity and health access by developing and testing differentiated models of HIV service delivery in high prevalence settings in collaboration with researchers from those settings. So on the left, you can see a number of my research team members who have contributed to the work I'll be presenting today. And as was mentioned, we use methods from a variety of different academic disciplines, including public policy, epidemiology, economics, biostatistics, psychology, and implementation science to address our research aims. On the bottom are the values of our team. We believe in research that is collaborative, high quality, will have high impact, is person-centered, is innovative, and we believe in shared learning. As many of you are well aware, daily oral pre-exposure prophylaxis, or PrEP, is one of the most effective HIV prevention interventions that we have, and thus delivering it at scale is important. In many settings, including Kenya, HIV incidence remains above levels of epidemic control and above UNAIDS targets. And PrEP has the potential to help reduce HIV incidence, but uptake in many high prevalence settings has remained low. PrEP is a public commodity in many settings in sub-Saharan Africa because it's donated by large international organizations like PEPFAR and the Global Fund. And thus, in those settings, PrEP is primarily being delivered in public healthcare facilities, specifically HIV clinics within those facilities, where a lot of barriers to client access remain, including stigma associated with visiting HIV, accessing HIV services when you're not living with HIV, limited hours of facility operation, and long wait times and travel distances to these facilities. To help achieve PrEP at scale, differentiated service delivery models could be utilized to help expand the reach of PrEP services and help ease access to those in need. The main principle of DSD is that a one-size-fits-all approach is not going to work for everybody, and that we need to differentiate the who, what, where, and when of how services are delivered to better meet the needs of different client populations and also lower the strain of health systems. For nearly the past decade, I've been working on developing and testing different models of HIV service delivery to help achieve these goals. For example, we recently completed, in collaboration with Dr. Jared Baton and Kenneth Ngure, a non-inferiority randomized trial that tested a model of 6-month PrEP dispensing supported with interim HIV self-testing to reduce the number of PrEP clinic visits in half. I also have a K99R00 award that's looking at testing a formalized model of peer PrEP referral enhanced with HIV self-test delivery to help increase PrEP initiation among adolescent girls and young women, which is a priority population in Kenya. And then we have a number of different studies that are focused on leveraging the private sector to deliver PrEP services in private pharmacies that are brick and mortar pharmacies, as well as private online pharmacies. And that'll be much of the work that I'm presenting here today. Getting PrEP into private pharmacies is a high priority for the Kenya Ministry of Health, which in an age of declining PrEP PEPFAR funding is really interested in leveraging the private sector to help sustain its HIV response. In the Ministry of Health's 5-year scale-up plan, they identified pharmacies as a target delivery point, and they identified pharmacy providers, including pharmacists and pharmaceutical technologists, as target prescribers. Additionally, this 5-year plan included a research agenda, and in this research agenda, they identified implementation research on PrEP delivery of private pharmacies as high priority. In Kenya and in other similar settings, private pharmacies are a promising study for PrEP delivery for a number of different reasons. First, in many low and middle income countries, roughly 50% of people first visit private pharmacies for their healthcare needs. Additionally, these pharmacies are well suited for addressing both urgent needs like STI treatment, as well as preventive care needs like blood pressure testing. It's also very common for individuals in these settings to purchase services at private pharmacies that are already available for free at public settings like contraception, for reasons including increased convenience. In Kenya, there's over 5,000 licensed pharmacies, and you can see the distribution of these pharmacies on the map on the far right. And these pharmacies are overseen by a regulatory board, and they have a formalized annual renewal process, and also have continuing professional development requirements for providers. In collaboration with Dr. Elizabeth Bukusi's team at the Kenya Medical Research Institute, as well as Dr. Kenneth Nguri's team at the Jomo Kenyatta University of Agriculture and Technology, we have been working on developing research around pharmacy-based models of PrEP delivery that are appropriate in Kenya for the past 5 years. This research has included formative qualitative research and stakeholder engagement to develop a care pathway for pharmacy-delivered PrEP services that is appropriate for Kenya. It also includes a number of pilot studies to test the feasibility of this care pathway. And now we're moving to starting a large 60 pharmacy cluster randomized controlled trial to test the effectiveness of different delivery models for PrEP in this setting, with the ultimate goal being that will generate high quality evidence that can be fed to policymakers that can help inform decisions around the scale up of this new PrEP delivery platform. In January 2020, we developed in collaboration with a number of Kenyan stakeholders from regulatory organizations and professional boards, a care pathway for pharmacy delivered PrEP services that was appropriate for Kenya. This care pathway was informed by qualitative research, and in collaboration with these stakeholders, we identified a number of challenges for delivering PrEP services following national guidelines in Kenya, and worked together to develop solutions to help overcome some of these challenges. So one of the first major challenges was PrEP prescribing. Pharmacy providers do not have prescribing privileges in Kenya. So what we did is we developed a prescribing checklist where pharmacy providers screened out individuals that reported any medical conditions that might contraindicate PrEP safety, and we referred them to public facilities for care by a PrEP clinician. Then another challenge was HIV testing. Pharmacy providers were not legally allowed to provide rapid diagnostic testing at private pharmacies, but they were allowed to provide provider-assisted HIV self-testing. So we got special permission from the Kenya Ministry of Health to initiate clients on PrEP using provider-assisted HIV self-testing, which went against sort of PrEP guidelines at the time. And then the last challenge was commodity procurement. So because PrEP is a public commodity, it's tightly regulated and controlled, and pharmacies don't have master facility list codes, so they're not able to distribute public commodities. So what we did is we developed a hub and spoke model where we paired private pharmacies with nearby public facilities, and then they received the PrEP commodities from those facilities, and whatever PrEP they dispensed was counted towards the sort of goals and the PrEP that was the PrEP dispensing goals for that facility. Here you can see the prescribing checklist that we developed as part of this care pathway that could be used at private pharmacies in Kenya. And this prescribing checklist was based off of one currently being used at the Kelly Ross Pharmacy here in Seattle to allow pharmacy providers to prescribe PrEP as part of their one-step PrEP program. We then conducted 2 pilot studies, and on the next slide, I'll talk about the differences between these 2 studies. But in both of these studies, they were both conducted in Kisumu, which is Western Kenya, and Thika, which is Central Kenya. And pharmacies had to be eligible for participation, they had to have a private room where HIV testing and counseling could take place. They had to be licensed and they had to have a full-time provider. And then for clients to receive PrEP services in the setting, they had to be over 18 years old and they had to meet the eligibility criteria on the checklist. To recruit these clients, we used posters, which we stationed around the pharmacies. And we also trained pharmacy providers to ask clients that were purchasing services indicative of HIV risk, like emergency contraception, if they might be interested in PrEP services. And then there was also a lot of informal word of mouth referral that occurred. All the PrEP drugs and HIV self-tests were given to us by the Kenya Ministry of Health. And our main outcomes were PrEP initiation, PrEP continuation, and a number of implementation outcomes, including acceptability. This chart here shows the differences between the two pilot studies that we conducted. The first pilot study was a small study funded by the National Institute for Mental Health. It took place at 4 pharmacies. We delivered oral PrEP. To be eligible, clients had to be over 18 years, meet the criteria on the checklist, and we excluded pregnant and breastfeeding women. We delivered provider-assisted oral fluid HIV self-testing, and all clients paid a 300 Kenyan shilling fee, which is roughly $3 for each pharmacy PrEP visit. We ran the study for 12 months, and we limited the number of clients that could initiate a PrEP at each pharmacy to 75 clients. Then we got funding from the Bill and Melinda Gates Foundation to extend this study and expand it to 12 pharmacies, which included the original 4. In addition to oral PrEP, we also added post-exposure prophylaxis, or PEP, to this model because we found there was a real need to that based on the high demand for emergency contraception in the study. And we also added STI testing at select pharmacies. We dropped the requirement that, or we enabled pregnant and breastfeeding women to access PrEP services in this extension study. We switched from oral fluid to blood-based self-testing. We dropped the fee to clients, so all these clients receive PrEP services for free. And we ran this pilot extension study for 6 months, and we did not put a cap on the number of clients that could initiate PrEP at each pharmacy. These photos here show pharmacy-based PrEP delivery in action. So you can see a number of the trained providers that participated in this intervention, as well as a number of the posters that, or the main poster that we used to help recruit clients in this setting. And you can also get a sense of sort of what these pharmacies look like in Kenya that we delivered this intervention in. In both pilot studies, PrEP initiation was high. So in the original study, where clients paid a 300 Kenyan shilling fee to initiate PrEP, amongst those determined to be eligible for pharmacy-delivered PrEP services, roughly 60% initiated PrEP. And in the pilot extension study, where we dropped this fee, among those determined to be eligible, over 90% initiated PrEP. And in both studies, the vast majority of clients that initiated PrEP in the study, in these studies, were PrEP naive and never previously used PrEP services. We then were interested in what the demographics of clients initiating PrEP in these settings looked like. And we found that roughly 50% were male, roughly 50% were under 25 years, and roughly 2 in 3 were unmarried. When we looked at the behaviors associated with HIV risk, the most common behaviors were having partners of unknown HIV status, having multiple sexual partners, which was over 50% of all clients initiating PrEP services in both pilot studies, and inconsistent condom use. We were then curious how these demographics of clients initiating PrEP at private pharmacies compared to those with clients initiating PrEP at public clinics in Kenya. So we compared the data from our pilot studies with that from a number of large implementation studies that have happened in Kenya, including the Partner Scale-Up Study, which delivered PrEP at HIV clinics, and the PRIA studies, which delivered PrEP in maternal and child health clinics and family planning clinics and focused on women. And when we did this comparison, we found that at the private pharmacies, we tend to be capturing a much younger population and a much higher percentage of unmarried people. And then when we looked at sexual behaviors, we also found that at private pharmacies, we tend to be capturing a population that has a greater percentage of partners of unknown HIV status and a greater percentage of partners of multiple sexual partners, and a much lower percentage of partners known to be living with HIV, which makes sense because especially at HIV clinics, a lot of the focus for PrEP delivery has been among serodifferent couples over the past few years in Kenya and other settings. These graphs here show what PrEP continuation looked like in both our original pilot study and our pilot extension. Over the duration of the original pilot study, 55% of clients returned to refill PrEP at any point. And in the extension study, this was 72%. Men over 25 years had the best PrEP continuation, and women under 25 years had the worst PrEP continuation, and clients over 25 years had better PrEP continuation than those under 25 years. Similarly, we were interested in how these PrEP initiation and continuation rates at private pharmacies compared to what is being observed within these implementation studies at public facilities. So in blue, you can see the levels of PrEP initiation and continuation in our pharmacy PrEP pilot studies. And in purple, you can see the levels of PrEP initiation and continuation in the implementation projects that are being done at public facilities. And additionally here, we've also added data from Jalinde, which was a large scale-up project run by Japaigo in Kenya. So you can see what these numbers look like. And while I would take the initiation number comparisons with a grain of salt, because the denominator is hard to be consistent for that one, when you look at the continuation metrics, you see the PrEP continuation at these private pharmacies was comparable, if not better, to what's being observed at public facilities in Kenya. In the pilot extension study, where we added PEP delivery, we found that PEP is also in demand at private pharmacies, and it may be a good gateway to PrEP. So among clients that were identified as being eligible for PEP services, which means that they reported a behavior associated with the risk of HIV exposure in the past 72 hours, we found that 95% of them accepted or initiated PEP. And among those that initiated PEP, 20% of them ended up transitioning and initiating PrEP later on. We also measured the perceptions of acceptability among pharmacy clients and providers in both pilot studies. And to do this, we use statements that assess different constructs of acceptability as outlined in the theoretical framework of acceptability. And we found that the vast majority of clients and providers agreed or strongly agreed with these acceptability statements. Here you can see a number of quotes that highlight the positive perceptions that both clients and providers had about pharmacy delivered PrEP services, including, I like that it is easily accessible and that the process is instant. It is easier to get PrEP here than at the hospital. And this will make the community that lives near a pharmacy know about the prevention measures that can help prevent the spread of HIV. Additionally, to help understand the sustainability of this model, we wanted to know what clients might be willing to pay for pharmacy delivered PrEP services and what pharmacy providers might be, pharmacy providers might charge clients for pharmacy delivered PrEP services if they were to receive the PrEP drugs and the HIV self-test for free from an organization like PEPFAR or the Ministry of Health. We found that in both pilot studies, including the pilot study where the clients received pharmacy PrEP services for free, the vast majority of clients were willing to pay something for each pharmacy PrEP visit. And the amount they were willing to pay was roughly 400 to 300 Kenyan shillings, which is equivalent to $4 to $3. And then pharmacy providers, they would be willing to provide this service. And they said they'd be willing to charge clients roughly around 200 Kenyan shillings, which is $2 per pharmacy PrEP visit, if they were to receive the commodities for free. And this helped inform the design of our cluster randomized controlled trial, where we tested different cost sharing models for pharmacy delivered PrEP services. So we have a 60 pharmacy 4-arm cluster randomized controlled trial that is hopefully will start in the next 2 weeks. And the team has been working very hard. I see Kendall and Pratika here to get this up and started. In this cluster randomized trial, the sort of control group that we have is what's currently within pharmacy providers' scope of practice at the moment if policy doesn't change, which is just identifying clients that are at risk and could benefit from PrEP services and referring them to public facilities for their PrEP needs. And then we have 3 different intervention arms. One where clients pay the fee for pharmacy delivered PrEP services, which we've set at 250 Kenyan shillings after a lot of formative research and engagement with providers and clients. Another arm where clients receive the services for free, but we, the implementers, will pay the pharmacist 250 Kenyan shillings for each pharmacy, for each client that they serve. And then we have a third arm where we're actually stationing an HTS counselor, an HIV testing and services counselor at the pharmacy to assist with counseling and HIV testing, which in the pilot studies, the pharmacists identified as sort of a weak point of this model because it takes a lot of time to complete the HIV testing and do the counseling, which prevents them from serving other clients and making profit off of those interactions. So the idea was to understand what additional benefit you might get if you station an HTS counselor at the pharmacy to assist with this model. Oh, and the main outcomes from this trial are PrEP initiation and continuation 60 days following referral. We also recently received some funding from the Bill and Melinda Gates Foundation to adapt this model to also deliver cabotegravir at private pharmacies, which is a long-acting injectable form of PrEP. And the objectives of this study is to determine the feasibility and additional benefit of delivering PrEP forms, including both injectable PrEP and the dapivirine vaginal ring at private pharmacies. We've set this up to be an interrupted time series design, which will start after we do some formative research with stakeholders to understand how we can most appropriately adapt this model. And we are going to do this at 8 private pharmacies. Before we can move to scale up of this intervention, a number of barriers remain, and we've been working in close collaboration with JPIGO, which is a large implementing organization in Kenya, to help identify and address some of these remaining barriers. So as we've discussed, one of the first barriers is what's currently within pharmacy providers' scope of practice at the moment, and pharmacy providers are not currently able to prescribe PrEP. So what we've been doing is we've been working in close collaboration with JPIGO and the Kenya Ministry of Health to develop training materials that are appropriate and could be used for scale of this intervention if the Ministry of Health chooses to pick it up. We've also been in discussions with the Kenya Pharmacy and Poisons Board and different pharmaceutical associations about incorporating PrEP into degree programs, training curriculums, and also incorporating PrEP delivery into continuous training, professional development opportunities for providers. Another challenge that remains is supply chain and financing of this intervention. So we've been in discussions with the Ministry of Health about giving pharmacies master facility list codes so they can deliver these public commodities. And JPIGO has also been really working very closely with the Kenya Ministry of Health to develop a framework for public-private partnerships, which could facilitate interventions like this in the future. And then finally, the last challenge is that around data and monitoring of PrEP distribution in these new private settings. So we've been working in close collaboration with Mesha Meds, which is a point-of-care pharmacy sales app that is used in over 3,000 pharmacies in Kenya. And we've been working with them to integrate our prescribing checklist into their point-of-care sales app, so that if this were to go live and go at scale, we're actually using Mesha Meds for our cluster randomized control trial. The Ministry of Health or another organization could get real-time data on the PrEP that's being dispensed in this setting. And Mesha Meds also has sort of a sophisticated auditing system that can help detect potential fraud for pharmacists that are delivering these commodities. So in summary, PrEP is not having the maximal impact on HIV incidence, and it's not easily accessible to all those that could benefit from it. Private pharmacies are a promising delivery channel that could help Kenya sustain its HIV response. And there's a lot of interest in this channel from a number of policymakers, including the Kenya Ministry of Health, the WHO, and funders such as Global Fund. And we actually recently heard that Global Fund wants to incorporate private pharmacies into its next round of funding as a PrEP delivery platform. So there's a lot of interest in scaling up this model. Our pilot studies have suggested that this model is feasible, it's in demand, it reaches populations that are naive, that are PrEP naive and could benefit from PrEP services, and that it's achieving outcomes that are comparable to those that are achieved at public clinics. However, there's currently no playbook on how to operationalize public PrEP services in private settings. So our studies are generating evidence that can help motivate this in terms of both effectiveness and costs. And as I mentioned, we're working in close collaboration with JPIGO to help address a number of the barriers to implementation and scale up. And then finally, Kenya is a great test case for the delivery of PrEP at these private pharmacies. And because often, if you can get something to work in Kenya and show that it's feasible, Kenya can serve as a great example for other countries throughout the region. And also, we think that this model could not only serve as a playbook for other countries, but in the future could potentially be developed or adapted for other interventions, such as cabotegravir, which we're hoping to do in the pilot study that I mentioned, as well as other combination prevention interventions or treatment interventions. So there's many people that I need to thank. I need to thank all the participating pharmacists and pharmacy clients that participated in the pilot studies, all the research teams that have contributed to this work, the Kenya Ministry of Health and Kenya Pharmaceutical and Poisons Boards, which have been great collaborators in the study, Daniel Ware at JPIGO, Nicole Young at the Gates Foundation, our funding sources. And then this is the pharmacy prep research team that is working on our cluster randomized controlled trial, which is hopefully starting in the next 3 weeks. And this was at a recent team retreat in Mombasa. So thank you, and I'm happy to answer any questions you have.
Our next speaker, CFAR seminar speaker for this month is Dr. Doug Kwon. Dr. Kwon has received his MD and PhD degrees from New York University and did internal medicine training at UCSF and New York Hospital, followed by ID fellowship at Mass General and Brigham and Women's Hospital. He's currently the Associate Professor of Medicine at Harvard and Director of Clinical Operations at the Reagan Institute of MGH, MIT, and Harvard. He has a clinical practice in the Division of ID at Mass General. And the focus of his research is understanding the role of mucosal tissues in HIV acquisition and disease progression, particularly in populations in sub-Saharan Africa. He's going to talk about his group's recent work examining the role of the vaginal microbiome on genital inflammation and adverse reproductive outcomes in women in South Africa. It's been a real pleasure hosting you. I know there's lots of overlaps and areas of interest that are very complementary with our groups here. So thank you for coming.
Yes, thank you. I mean, it's great to be here in person because like you said, I think a lot of seminal work has been done here at UW and Fred Hutch in particular. And so it's great to kind of visit with people and actually meet in person once again. So I'm going to start talking about a little bit of the background work that we did and then kind of move to like a number of kind of unpublished stories. But first to kind of contextualize the work that we've been doing, looking at the vaginal microbiome and its role in HIV risk. I think this audience fully understands that there's clearly an unequal burden of HIV, particularly in sub-Saharan African countries where most new infections are occurring. And within sub-Saharan Africa, the unequal burden on young women of reproductive age in particular, where HIV prevalence rates are considerably higher amongst young women compared to many young men. And as Katrina kind of talked about, there are a number of different methods to actually prevent HIV infection that are effective when taken, but also we've seen that in a number of randomized controlled studies that women in sub-Saharan Africa actually have had a harder time using these methods to prevent their own risk for HIV. So we've been involved in a study in Durban, South Africa, in this township called Umlazi, called the FRESH study. Oh, yeah.
Can we hide the
Oh, that's not going to do it.
It's a presenter mode, so we're not able to.
We're just drag it.
And again, you're supposed to go. Oh, yeah.
Okay. All still learning how to use Zoom. We've been involved in a study called FRESH, which stands for Females Rising Through Education, Sport, and Health. This study has enrolled over 2,200 HIV uninfected young women in this township of Umlazi, just outside of Durban. The participants come twice a week. They get tested for HIV at each visit through a finger prick viral load PCR. They also have regular pelvic exams and blood draws at 3-month intervals. And they're engaged in a twice-weekly class that focuses on poverty alleviation and self-esteem, which requires them to stay there for 6 hours of class each week for 10 months. And I think one of the things that we're very proud of is the fact that most of the participants go on to some sort of meaningful activity afterwards, which is a job or internship or further schooling. And yet the HIV incidence rates remain enormously high and really unchanged from the community. And just to kind of give you a sense of sort of what this looks like, in this region of KwaZulu-Natal, there are very high rates of HIV incidence and prevalence amongst young women, where in a population study, the Vukuzazi study, in the 20s and 30s, HIV prevalence rates amongst young women in their 20s and early 30s exceeded 60%. And I think as the prior speaker talked about, we've looked at PrEP within FRESH. What we've seen is that although everyone in FRESH are offered PrEP with no charge, only about half of the women actually book first appointment to initiate PrEP, and then only about a third of all the participants actually show up for the first appointment to get their first month of PrEP. And then there's a very high attrition rate where you lose about half of the women each month, so that by the time the third month, only 7% of the women are actually continuing on PrEP. And so, there are a lot of challenges clearly to sort of initiating PrEP in this this setting. And so maybe you can ask for your help and figure out how to solve this problem. But I think that there still are opportunities based upon the biology to kind of create interventions to try to reduce HIV acquisition. And one of the things that we know is that when women are exposed to HIV through heterosexual sex, they're exposed to enormously diverse quasi-species of viruses within the mucosal tissues of the female genital tract. And in most cases, it's thought that a single virion is able to kind of break through these intrinsic barriers to infect a local activated CD4 T cell within the tissue. And that then there's this initial period of obligate expansion there within the tissue before you have a sufficient burden to actually then disseminate and sort of establish this systemic latent reservoir. And that initial period that's localized within the tissue is thought to take about 1 week. And so there's a potential opportunity there to intervene to kind of reduce, to stop that dissemination and prevent establishment infection. And so we've been really interested, as others, in sort of what are the mucosal factors that affect the efficiency of establishment of infection. And one of the things that was really actually done early on by Joanne, who's here, and others at Caprisa was a retrospective study of the Caprisa 004 trial, which is a vaginal microbicide gel that contained the antiretroviral tenofovir. And in this study, post hoc analysis, it was discovered that women who had higher levels of inflammatory genital cytokines actually had an over 3-fold higher risk of becoming infected with HIV. And so I think as soon as Joanne had kind of done this really sort of seminal work, we looked within FRESH to sort of see what the inflammatory cytokines look like. And we saw this very wide range of baseline inflammatory cytokines in these women who were otherwise clinically asymptomatic and healthy. And we saw that women in this highest quartile inflammatory cytokines, and in this representation, each column is a different FRESH participant, each row is a different cytokine. This is shown on a log scale so that women in this highest quartile had up to 1,000-fold higher cytokines than women in the lowest quartile. We looked to see if there was any clear cause of that high genital inflammation. And we thought maybe STIs might be a cause, but in fact, only about 25% of the women with high levels of baseline inflammation actually had an STI. And there were no other associations with other factors like behavioral factors, sexual behaviors, contraceptive use, or demographics. So we looked at the vaginal microbiome as potentially being a mediator of bacillal inflammation. And so this is sort of what the kind of initial profiles looked like, where each column is again is a participant in FRESH, and each color is a different bacterial taxa. And what you could see is that most of the women actually had communities that were comprised of mixed anaerobes. And so we called each of these communities cervicotypes, or CT. And so CT3 and 4 were both these mixed communities that had very little lactobacillus and had high diversity anaerobic communities. CT1 were communities that comprised less than 10% of the women, and those were women who had a predominantly Lactobacillus crispatus dominant community, which sort of we kind of typically think as the most optimal. And then about a quarter of the women had a community that we called CT2 that was dominated by a different species of lactobacillus called Lactobacillus inners. So what we showed was that women with these high diversity CT3-4 type communities had much higher levels of inflammatory cytokines. They had higher numbers of activated CD4 T cells within the tissues of the cervix. And again, those are thought to be the first cells to be infected by HIV. And so we thought that could be one biological mechanism by which these women would be at higher risk for acquiring HIV. And when we looked prospectively, we saw that, yes, these women with CT3-4 type communities with these mixed anaerobes were acquiring HIV higher rates, up to 4-fold higher rates than women with a Lactobacillus crispatus dominant community. And this work paralleled very nicely work that has been done here, looking at vaginal bacterial concentration. So David Fredericks and Sujatha and Scott McClellan and others here actually did this very nice study, now looking at quantitative measures of vaginal bacteria, and showed that there were quantitative associations with 7 different bacterial taxa and HIV risk across 5 different cohorts of women within sub-Saharan Africa. And one of the things that I think a lot of us have been looking at now is sort of like, okay, we have these associations between vaginal bacteria, inflammation, and HIV risk. What are some of the mechanisms by which that that increased inflammation is being mediated. So we've done some simple studies looking at transcriptional analysis of cells from the female genital tract in women who had lactobacillus crispata dominant communities versus these mixed anaerobic communities, and shown that there are multiple different cellular lineages, including epithelial cells, T cells, neutrophils, but in particular, cervical antigen presenting cells seem to be a really important population for sensing these communities. Those cells have the most differentially expressed genes and gene pathways, comparing women with CT3/4 type communities versus CT1. And one clear signal of signaling there is TLR4 signaling or LPS signaling. LPS is a component of gram-negative bacteria that's highly enriched within these anaerobic communities. And so that clearly is one mechanism that sort of is being used to sense these more inflammatory bacterial communities. And it was interesting because we saw that a lot of these pathways that are upregulated in women with these CT3/4 type communities had also been described as being associated with things like preterm birth and preterm labor. Things like IL-1 beta, TNF signaling, LPS signaling, NF-kappa B pathway activation, IL-8. Those had previously been described as being associated with preterm birth, but not in the context of sort of microbial differences. And so we think that actually there's a lot of, that this overlap in signals indicates that, you know, that there may be common pathways by which vaginal bacteria may be contributing to both of these outcomes, HIV risk and preterm birth. And I think that over time, there's been a more consistent emerging picture where these vaginal bacterial communities may be impacting a lot of women's health outcomes. So in particular, having lactobacillus non-inners dominant microbiota are associated with lower rates of preterm birth, lower rates of infertility, lower rates of cervical dysplasia, better success with in vitro fertilization, and lower rates of other STIs than HIV. And many of these problems have higher burden within low and middle income countries. And I think this sort of has captured our interest in sort of hoping that there might be a way to kind of leverage this understanding to address not just HIV, but multiple different women's health outcomes. But one problem has been always like, how do you get to mechanistic understanding, right? And a challenge has been that, you know, there are not really good models within this field of sort of that capture vaginal bacteria and kind of host read and kind of all the necessary host components, which include probably epithelial cells as well as immune cells. And so one of the things we've looked at is whether or not a humanized mouse might be useful here. And so we've used these mice, which are called BLT mice. They basically are mice that lack an intrinsic adaptive immune system that are reconstituted with human stem cells and then can be infected with HIV. So what we then did was to kind of take specific vaginal bacteria that are associated with increased HIV acquisition risk and increased genital inflammation in women in fresh, culture out isolates of those bacteria. And we kind of sought to see, we tried to see if we could actually then vaginally inoculate these mice and see if those bacteria would persist. And so one of the things that we saw was that you can establish colonization for a period, but that quickly goes away. So when you vaginally inoculate with Lactobacillus crispatus or Sneathia sanguinigans or Prevotella bivia, these two bacteria were associated with increased risk, that they would dominate the community for less than a day. And then basically they would revert, the communities would revert to sort of the natural endogenous mouse community. But by doing repeated vaginal inoculation, you could see and recapitulate some of the observations that we saw in women, namely that anaerobes like Sneathia and Prevotella were associated with a number of increased levels of genital cytokines, the same ones that we saw elevated within women in fresh. There were more CD4 T cells within the tissues of the genital tract. And if you did a repeat HIV low-dose vaginal challenge model, where you would basically weekly inoculate with specific vaginal bacteria and then expose the mice to a low-dose HIV vaginal challenge, you could see over time that the mice that were inoculated with Prevotella actually acquired HIV at higher rates than mice that were vaginally inoculated with lactobacillus. And the mice that were vaginally inoculated with Sneathia sort of had a middle phenotype where there was a trend towards increased HIV acquisition, but that wasn't significant. But it was interesting because it sort of modeled what we saw when we looked at inflammatory cytokines, where mice that were vaginally inoculated with Sneathia had sort of elevated but moderately elevated inflammatory cytokines in the genital tract relative to Prevotella. So, the question is, so we have a model system that recapitulates some of the expectations based upon our findings in women in fresh. Can we now use this to sort of dissect further mechanistic questions? And so we're hopeful about using this model despite its caveats. And we think at the very least, it kind of supports, a directionality between these associations where we think that these inflammatory anaerobes in the vagina are driving post-inflammation that then sort of impacts these reproductive outcomes. And so I think that kind of framework gives us hope that if we can sort of better understand how this inflammation is being mediated, we can hopefully intervene to try to reduce these poor outcomes. But we wanted to sort of better understand how these host microbiota interactions more specifically sort of are occurring. And one thing that we've been interested in is sort of looking at helper T cell responses. And we know that, for those of you who are immunologists, we know that CD4 positive T helper cells really have critical roles, especially within tissues of sort of modulating the inflammatory environment. And there are different lineages of T helper cells. For example, Th1 and Th17 cells actually can produce a more inflammatory environment, whereas Tregs are sort of more homeostatic or sort of immune regulatory. And another thing that we've been really interested in sort of the kind of literature that's developed in the gut, where particularly in mice, what's been shown is that gut bacteria can very kind of precisely differentiate and skew T helper subset differentiation to sort of more inflammatory or less inflammatory types. So for example, I think in work that's been really sort of groundbreaking, it's been demonstrated that if you monocolonize germ-free mice with segmented filamentous bacteria, the single bacterial taxa, you can actually skew the differentiation of T helper subsets towards pro-inflammatory Th17 subsets. Whereas if you use other types of bacteria, like in this case, Listeria, you can actually skew towards Th1 phenotypes. And people have shown that, for example, if you look in humans and patients with inflammatory bowel disease, you can see sort of E. coli specific responses that in inflamed mucosa, you can skew towards these T helper subsets that have particular profiles that are pro-inflammatory, and that these commensal specific T cells actually are reduced during periods in which there's less inflammation. So the question is this same kind of thing happening in the female genital tract? So the first characterization we've done is to kind of look at the clonality of T cells within the female genital tract. And if you look at paired blood and T helper subsets in the cervix, what you can see is that there are these very highly expanded clones within the genital tract. So some of these clones are occupying 10% or more of the repertoire. So that's a huge degree of expansion relative to matched blood, where where you actually rarely see clones that are highly expanded. And if you can kind of look at this in a variety of ways. If you just look at sort of the diversity index, you can see that there's much less diversity in the cervix than in the blood between matched individuals. Or if you look at sort of the D50, sort of the number of clones that it takes you to hit 50% of the TCR clonal population, you can see that those numbers are much lower in the cervix than in the blood, meaning that the clones are much more highly expanded in the cervix than the blood. So you have these highly expanded clones within the genital tract. These clones are actually also represented in the blood, but at much lower frequencies. So for any clone that's highly expanded within the cervix, you'll see actually a correlation with that same TCR clone in the blood, but at much, much lower levels, right? So they're there, but at least a log less. And when you do see them, what you see is that if you see sort of a highly explanted clone that's of the Th1 helper lineage in the cervix, that when you look in the blood, its presence is primarily within that same lineage within the Th1 population. For Th17s in the cervix, you can also see like primarily when they're in the blood, they're in the same lineage, and similarly for Tregs. So the question is like, so you have these highly explanted T cell clones in the genital tract. What are they directed against? And are they directed against commensal bacteria? And so we started to look at this by kind of stimulating with pools of vaginal bacteria that are actually cultured from women in fresh, and look to see whether or not women in fresh actually can mediate responses against some of these bacteria. And what you can see is that there are responses against bacteria, but at low levels. But there are some reasons to think that these actually may be real. For one, that they're primarily occurring within the CD45RA negative population, which is a memory population, which one would expect. That there's a broad range within different individuals of responses to different vaginal bacteria, but these do seem to be consistently mediated by interactions with MHC class 2. So if you use a class 2 blocking antibody, you can see that you sort of reduce all of these responses. And that means that these responses are mediated through the TCR likely through kind of class 2 interactions, which also makes sense. But how do we actually define really the specific cognate epitopes that these T cells recognize? And for that, we're basically now kind of making TCR reporter lines where we can clone in the expanded clones, TCRs, the matched alpha and betas, into cell lines and then stimulate those cells with different antigens that we actually have been working with Romnick Xavier's lab using computational methods to sort of predict what are the bacterial antigens that are most likely to be presented based upon that patient's HLA. And so we're now kind of going through these kind of TCR reporter lines to sort of see if we can define the specific bacterial epitopes. We've also been kind of using some additional novel methods through a collaboration with Michael Birnbaum, where he's able to engineer VSVG viral particles that have on their surface MHC class 2 with peptide libraries. And what you can do then is that that virus will specifically recognize a T cell that recognizes that particular bacterial epitope and then infect that cell and then turn on a reporter. So you can recover clones that way by screening them for those that specifically recognize that bacterial peptide within the context of class 2. And then you can just sort all those T cells that actually light up and then clone the TCRs and then can make cell lines to again confirm that those are actually truly specific for the targeted epitope. So that's kind of one way that we're looking at this. And we have other things that we're doing on the host side, but we're also, I think, really interested in kind of understanding vaginal community dynamics and seeing how that might inform strategies to kind of alter the vaginal microbial composition. And one of the things that we've seen looking at sort of longitudinal sampling in fresh is that these communities can be dynamic, and that when they do transition, that there's this general pattern where if you start out with Lactobacillus crispatus, you can transition to one of these anaerobe dominated states, CT3 and 4. But when you do so, you almost always go through an intermediate state that's Lactobacillus inners dominant. And then once you're in this Lactobacillus inners dominant state, the CT2 state, you have high probabilities for transitioning to these CT3-4 type communities. So we found that really interesting, and we kind of, based upon these sort of transition probabilities and the models that sort of inform them, we kind of asked this question like, okay, if that's the case, then what kind of interventions would you want to design to shift the equilibrium state so that more women are in CT1 Lactobacillus crispatus dominant communities? Because like I said, in the fresh study, less than 10% of women have those communities, even though those are associated with low inflammatory states and lower risk for HIV. And based upon those models, it's a little bit of a non-obvious answer, which is that the transition probabilities that you want to target are the transitions from, you want to reduce the rates of transitioning from crispatus to inners, or you want to boost the rates of transitioning from inners to crispus. And only by doing that can you actually achieve transition states that increase the amount of lactobacillus crispus that you have at equilibrium. That's kind of designated by this orange color. If you target any of the other possible combinations of transition states, they're not very effective at actually shifting the equilibrium towards more lactobacillus inners, lactobacillus crispus. And this was interesting to us collectively because it kind of suggested why treatment for bacterial vaginosis might be so ineffective, where you have recurrence rates of over 50% at 6 months. And it's because multiple studies have shown that when you treat women with bacterial vaginosis with metronidazole, you kind of start out with these communities that are anaerobe dominated with very little lactobacillus, and then afterwards you highly enrich for lactobacillus inners. That's been known for a long time. We've been doing some analysis in a clinical trial, which I'll talk about later, but you can kind of see that as well here, where you start out with BV, and these women with BV, they get treated with metronidazole, and you have this large shift towards lactobacillus inners dominant communities. And then sort of after you stop treatment, there's this sort of slow shifting from inners to these sort of BV-like communities again. So, part of the problem may be that current interventions with antibiotics shift these communities from anaerobe dominated to lactobacillus inners, which I said has this high probability of transition back to BV. And so the question is, like, can you actually create interventions that shift you all the way to lactobacillus crispus rather than lactobacillus inners? So that led us to be very interested in sort of understanding better the biology of lactobacillus inners. But the problem in the field was that lactobacillus inners is generally poorly studied, in part because it's been very difficult to grow it in vitro. And sort of there have been few cultured isolates to actually study. And so lactobacillus inners is really interesting because it's the most prevalent vaginal bacterial species worldwide. 25 to 40% of women worldwide have a lactobacillus inners dominant community. Lactobacillus inners is really interesting microbiologically because it expresses putative virulence factors. It also has like a very small genome size, suggestive of real kind of niche adaptation. It's been associated with various sort of poor outcomes, including preterm birth and HPV risk and cervical dysplasia. And as I said, it's been classically difficult to grow because it doesn't grow in standard media. So we had a postdoc in the lab who sort of took on the problem of trying to figure out how to get lactobacillus inners to grow. And to summarize a lot of work that he did, he discovered that if you actually supplemented media with cysteine, that you could now get lactobacillus inners to grow in vitro. And cysteine supplementation didn't sort of affect the growth of lactobacillus crispus or anaerobes like G. vaginalis. And that allowed him to then kind of quickly accumulate a library of lactobacillus inners strains from globally diverse sources. And then he kind of selected, based upon sort of phylogenetic breadth, certain isolates to then test. And pretty consistently, you could see these augmentations of lactobacillus inners strain growth when you supplemented media with cysteine. So that was good in a practical sense because that now allowed us to actually like study lactobacillus inners. And so one of the things that we wondered was, well, why does lactobacillus inners need exogenous cysteine to grow? And one initial hypothesis was that, oh, maybe lactobacillus inners just lacks the intrinsic machinery to create cysteine, but other lactobacilli have that machinery. And so cysteine is generally kind of made through sort of canonical pathways, which are sort of the cysteine synthase pathway or this reverse transsulfuration pathway. And when we look genetically at hundreds of different strains of various species of lactobacillus, what we saw is that they all pretty much lacked intact biosynthetic pathways for making cysteine. So that wasn't the answer. None of them could really endogenously make their own cysteine. But when we looked in vivo, what we did see was that there was a very close correlation with lactobacillus levels within the genital tract and cysteine levels. So if you look at women who had Lactobacillus crispatus or Lactobacillus inners dominant community, they had high levels of cysteine relative to women who had low levels of Lactobacillus inners. And this was true when you did kind of like a correlative analysis. And you could see, again, lactobacilli highly enriched in women who had high levels of cysteine. So we thought that cysteine was important for these lactobacilli levels in vivo, but the question was, why do we see this differential growth effect? And so one of the ways that bacteria utilize cysteine is not just by uptaking cysteine, but uptaking cysteine-containing molecules like glutathione. And there are dedicated cysteine transporters, generally by thiol transporters, that basically uptake these more complex cysteine-containing peptides, such as glutathione. And so when we looked at transport mechanisms for glutathione transport, what we saw was that other strains of lactobacilli other than inners actually all consistently had these transporters, but Lactobacillus inners uniquely lacked this glutathione transporter. And if you looked at labeled cysteine uptake and glutathione uptake, what you saw was that both Lactobacillus crispatus and inners could take up labeled cysteine, but only Lactobacillus crispatus could uptake labeled glutathione, whereas Lactobacillus inners couldn't, which means that the reason why that Lactobacillus inners requires exogenous cysteine is because it lacks the ability to kind of take up these more complex molecules that contain cysteine, whereas other lactobacilli can take those up. So that gave us some insight into the basic biology, but then this question came back to like, could we possibly leverage this to think about a way to, again, treat these BV-like communities to shift them all the way to Lactobacillus crispatus rather than leaving them at Lactobacillus inners. And so what was needed there was like some intervention that could specifically suppress Lactobacillus inners growth, but allow Lactobacillus crispatus growth. And so we tried these substances which are cysteine uptake inhibitors, and in this case, it's one called S-methylcysteine or SMC. And what we saw when we cultured bacteria in vitro was that, in fact, that's what would happen. Whereas if you added increasing concentrations of SMC, you didn't affect the growth of Crispatus, Jensenii, or Gasseri, all Lactobacillus species that are thought to promote better health outcomes, but you could suppress the growth of Lactobacillus inners. And so we tested this idea like, okay, could you use something like SMC plus metronidazole to actually then shift communities towards Lactobacillus crispatus dominance? And at least in vitro, you could, where if you took these mixed communities that were comprised of Crispatus, inners, and anaerobes, by combining both metronidazole to get rid of the anaerobes and SMC to shift from inners to Lactobacillus crispatus dominance, you could actually sort of greatly increase the abundance of Lactobacillus crispatus. And so that's one possible method to try to get better clinical outcomes. But what about other ways to durably alter the vaginal microbiome? So I think this is also something that we've been interested in. And I think one kind of thing to sort of point out about this is that, you know, what about vaginal probiotics? We hear a lot about sort of probiotics for the gut. You know, are vaginal probiotics effective? And clearly there's a need for better interventions. You know, as I mentioned, you know, there's recurrence rates of BV that are sort of above 50% after 6 months of treatment with antibiotics. Bacterial vaginosis is, is present in 20-30% of all women worldwide, and there's been no change in recommendations for treatment of BV in the past 40 years. So what about, if you go to Whole Foods, you'll see a whole shelf full of vaginal probiotics. Like, are they effective at all? And the problem with most of those probiotics are that most of them are actually formulated with dairy or animal or gut lactobacilli, but not lactobacilli that naturally colonize the female genital tract. And in what limited studies there have been, there's been very little efficacy demonstrated. So, although there has been this one trial of a product called Lactin D, which contains a single strain of Lactobacillus crispatus called CTB05, which was actually isolated here by Sharon Hillier and others from a female African-American donor. This is administered as a freeze-dried powder via a vaginal applicator, and it's given at a dose of 2 × 10⁹ CFU. And in this study that was published in the New England Journal in 2020, what they showed was that women who received this Lactin-V product after treatment with antibiotics for BV had a 50% reduction in BV recurrence at 12 weeks. So this product was, so basically women with BV were enrolled in the study, they were treated with topical metronidazole for 1 week, and then they were treated with Lactin-V or placebo for 11 weeks. And then the primary endpoint was week 12, basically right after they stopped. And then there was a secondary time point at week 24, 3 months after stopping. And you could see that there was this 50% reduction, although recurrence rates were still quite high. In the placebo group, it was 45%, and that was reduced to 30% at 12 weeks when you basically have just finished your course. And that difference decreased as you went 12 weeks after that. So there was some loss of durability there over the 3 weeks after you stopped treatment. So we've been looking at the results of this study, but these kind of results were based on clinical endpoints of a combination of Nugent score plus AMSL criteria, so clinical criteria to diagnose recurrent BV. So what about sort of using molecular methods by bacterial sequencing to kind of look at the results of that study? So we basically did sequencing. We did 16S sequencing. We also did metagenomic sequencing. We kind of assessed inflammatory cytokines, and then also combined that with all kind of the very rich kind of demographic and behavioral data. And what we sort of define as sort of a measure of successful Lactobacillus crispatus colonization were very simple metrics. You know, did you achieve greater than 50% Lactobacillus crispatus, you know, at week 12? And so this is what kind of greater than 50% Lactobacillus crispatus looked like within the study. Did you achieve greater than 50% total lactobacilli? So that's these two groups combined. Or did you have less than 50% total lactobacilli, which are basically what this kind of looks like, which again are like mostly communities that are dominated by mixed anaerobes. And when you kind of compare these simple definitions against sort of the clinical BV scores in the trial, they sort of generally coincided with expectations, where these communities that were lactobacillus dominant had, you know, basically no people who were diagnosed with BV, and then these kind of women who had less than 50% lactobacillus had sort of mixed diagnoses of BV or non-BV. And so what did the studies, what did the outcomes look like when looking at these sort of molecular endpoints? What you saw was that at week 12, which again, it was right after Lactin-V was stopped being administered, you saw certainly more Lactobacillus crispatus, which is sort of in this kind of yellow gold color. But you still saw a good amount of Lactobacillus inners, whereas in the placebo, you had actually very little Lactobacillus crispatus, and mostly the non-lactobacilli communities were dominated by Lactobacillus inners. This is looking at a Sankey chart, but if you look at sort of the stacked abundance plots, sort of this is what it looked like at week 12. So again, clearly, like by eye, you can see that there are more women with Lactobacillus crispatus dominance in the Lactin-V group than in placebo arm. And if you kind of looked at sort of the overall outcomes, what you saw was that there was 3 times more Lactobacillus crispatus dominance at week 12 in the Lactin-V arm than the placebo arm. And then at week 24, that actually increased to over 4-fold. So what's interesting is what seems to be happening is that the reason why that's increasing is because at the difference between week 12 and 24, you basically maintain most of the women who have Lactobacillus crispatus at that time point are maintained, but you're losing more of the women here who have Lactobacillus crispatus, you know, at week 12 in the placebo. And at week, if you look at the endpoint of total lactobacillus, what you see is that there's actually no difference at week 12. Again, but keeping in mind that most of these women in placebo have lactobacillus iners dominance, while most of the women in the lactin D arm have lactobacillus crispus. And then you start to see a difference at week 24, again, as more of these women in the placebo arm shift from lactobacillus iners to sort of these BV-like communities. So I think that, by that measure, there was, a high degree of success, but still a lot of room for improvement. So the next thing that we wanted to look at was sort of kind of looking at strain variation within lactobacillus crispus. So using the metagenomics, we could look at sort of, okay, of these women who established greater than 50% lactobacillus crispus, was that always the CTV05 strain that was in the product? Or were there other lactobacillus crispus strains that were colonizing? And so we basically used a SNP calling approach where we could look at single nucleotide variants between the different lactobacillus crispus strains. And what we saw is that at that week 12 time point, most of the women who had lactobacillus crispus were colonized with CTV05. But there were 15% of women who actually had a strain that was not CTV05. And by week 24, that actually became 30% of women who had a non-CTV05 lactobacillus crispus strain. So that was pretty high. I think we were surprised by that, like how many women seem to be colonizing with a non-CTV05 lactobacillus crispus strain. And so looking at sort of the strain dynamics kind of on an individual scale, these are just some examples where this is kind of the amount of CTV05. You can see, for example, these two participants had CTV05 throughout. They sort of colonized early with that strain and stayed colonized. Looking at sort of a correlation matrix here, you can see that this is kind of this blue block here, women who had CTV05. But there are others who actually started with CTV05, and then their lactobacillus crispus was overtaken by a different strain. Or in this case, this is a participant who actually colonized with lactobacillus crispus, but a non-CTV05 strain. And you can kind of see that this woman who's designated by these light blue blocks is here, and sort of she's being colonized with this strain of crispus, which is outside of this CTV05 block. So we're really interested in why that might be. And so we're looking at sort of what are the different kind of genes and gene pathways that are present in these non-CTV05 colonizing strains, and seeing if there might be clues in terms of why they're able to colonize. And also we're really interested to sort of see if they actually might be present at low levels in that woman's community at earlier time points, and maybe somehow overtaking the CTV05 strain once lactobacillus crispus is established, dominance is established. We also looked at kind of how this intervention affected general inflammation with this hypothesis that treatment would reduce general inflammation. And overall, we saw sort of an expected correlation of cytokines where when women had less than 50% lactobacilli, they had higher levels of inflammation. And when they had greater than 50% lactobacillus crispus, they had the lowest levels of inflammation. And overall in the trial, if you looked at both the lactin B and placebo arms, you saw a significant decrease in inflammatory cytokines with treatment, which was probably effect of just the antibiotic treatment, not necessarily the CTV05. And between the two arms at week 12, there was no difference. But then you saw a difference at week 24, again, as more of these women shifted from lactobacillus iners to sort of these more diverse communities. And so we wanted to kind of understand what were the factors that were associated with successful response to the intervention. So, what we saw was that 28% of lactin B participants had colonization with lactobacillus crispus at week 12. That was over threefold higher rates than in the placebo arm. And so can we kind of identify factors that explain those who benefited from the intervention? And so here we kind of leverage a lot of the metadata that we had, including kind of the biological metadata, but also behavioral data, to sort of see, okay, what things were predicting successful colonization at that week 12 time point. and we could kind of leverage all these different data endpoints, but particularly focusing on those things that were either present before treatment with metronidazole or after treatment with metronidazole. And so this was integrated into a model by Laura Simel and Susie Holmes at Stanford, using what's called a multi-block analysis, where you can kind of fit a model that basically blocks possible explanatory variables into thematic blocks. And so there are things like, which arm were you in? Were you in the lactin B arm or the placebo? Demographic factors such as age, race, methods of birth control, different microbial communities that start, the presence of different cytokines, your total bacterial load after treatment, vaginal hygiene practices, or sexual behavior. And using that analysis, what you saw fall out was that certainly if you were in the lactin B arm, you are more likely to colonize with Lactobacillus crispus. So that was just reassuring. That made sense. That gave us confidence that what we were doing, jived with kind of our expectations. If you were Black or African American, you had a lower probability of colonization with Lactobacillus crispus. And there was a trend towards white women having a higher degree of colonization with Lactobacillus crispus. Also, there were certain cytokines. For example, low levels of NIG or higher levels of MIP1 alpha or IL-1 alpha before treatment with metronidazole that predicted successful colonization. Various types of sex during administration of the treatment were associated with decreased chance of colonization. And one of the greatest factors was sort of your total bacterial load as determined by 16S qPCR after metronidazole treatment. So there is a very clear correlation, negative correlation between your 16S bacterial load after treatment with metronidazole and crispus colonization. So the greater the reduction or the lower the levels of total bacteria after treatment, the higher your probabilities of colonizing with Lactobacillus crispus, suggesting that you kind of needed to sort of clear the field to sort of make space for this exogenous kind of probiotic to colonize. So kind of the summary of the lactin V clinical trial results were, so the lactin V recipients were significantly more likely to colonize with Lactobacillus crispus at week 12. And that increased by week 24 to greater than 4-fold more likely. But greater than 50% of Lactobacillus crispus was achieved in less than half of the lactin B recipients. So there certainly is more room for improvement. Many of the lactin B recipients were colonized with the CTV05 strain. But I think, to me, somewhat surprisingly, is that a number of women weren't. So 30% of women who were colonized with Lactobacillus crispus did not have significant amounts of CTV05. This association of having these specific inflammatory cytokine signatures prior to BV treatment as being associated with Lactobacillus crispus colonization is something that we're kind of interested in looking at and wondering if that may have some implications about like the type of host response that you mount against these BV-like communities as having a role in whether or not you can successfully displace those communities. And then that the total bacterial load after metronidazole and some racial factors seem to be inversely correlated with successful colonization. So, and then kind of, again, sexual activity being negatively associated with colonization. So that's kind of, I think, some of the things that we've seen from the lactin V trial, which was a trial of a single strain of Lactobacillus crispus. What about next generation interventions that could be tried? So one of the things that we're involved in with the Gates Foundation is kind of creating more kind of rationally designed and geographically diverse consortia of Lactobacillus crispus from women from different regions of the world that have stable Lactobacillus crispus dominant communities over time. And so in collaboration with David Roman at Stanford and Jacques Rebel at University of Maryland, Joanne at University of Cape Town, Sanae Nagapu at Caprisa, and sort of some of the isolates that we've gotten from Fresh, we've done sort of isolate collection with those collaborators to kind of create kind of collections of geographically diverse isolates. And one of the things when you look at those genomes of isolates, there are actually significantly different genes and gene functions across the African and North American isolates. And so using kind of a method that was developed by Jacques Rebel and Michael France, we then kind of selected consortia that try to recapitulate the genetic breadth of metagenomes of all the genes that are present in these stable Lactobacillus crustatus communities. And so using that, the consortium has come to sort of the VMRC, the SCATES project has come to 2 specific consortia, one that's comprised of 6 different geographically diverse strains of Lactobacillus crustatus, and another that contains 15 strains. And so that is going to go into trials this summer in July in both the US and in South Africa. The other thing that we've been involved in with Carolyn Mitchell, who's an OBGYN at MGH, is vaginal microbiota transplant, or VMT. So this is akin to FMT, where people in the gut world have taken fecal samples from healthy people and try to introduce them into the gut to displace dysbiotic gut communities, with the idea that, you know, it may be, especially if you have certain fragile communities, it may be easier to kind of displace that whole ecosystem than trying to use probiotics that have like a finite number of strains. And so we started this VMT trial in Boston, which randomizes women to either receiving VMT, which are secretions from healthy women with Lactobacillus crustatus dominant communities, or placebo after metronidazole, to sort of see how successful that approach might be within women with a history of recurrent BV. Okay, so just to summarize, you know, I think that we think that there are a lot of really important effects on the mucosal environment in the genital tract that are mediated by these specific cervical vaginal bacterial communities, and that in general, having a Lactobacillus dominant community, particularly one dominated by Lactobacillus crustatus, seems to be beneficial in that it seems to sort of mediate a low inflammatory state where you have a lot of these protective barriers, such as mucus and secreted immunoglobulin intact, but that when these communities are replaced by these diverse communities with anaerobes, you have a lot of breakdown of these natural barriers, including damaged epithelia, induced inflammation, breakdown of mucins and mucus, cleavage of immunoglobulins, formations of biofilms, and other and more soluble and cellular measures of genital tissue inflammation, and that those collectively, by yet unknown mechanisms, then contribute to a number of adverse reproductive outcomes. Okay. So I just kind of want to thank all the people who have been doing the work, including a lot of our collaborators, of course, the folks at Fresh, folks within the VMRC, including Jacques Rebel at University of Baltimore, Susan Holmes, David Relman, and then of course our collaborators at Caprisa, including Joanne, and happy to take any questions.
Great talk.
Thanks.
There's so many wonderful threads there. In terms of the lactin B story,
Yeah.
it seems like Lactobacillus crustatus begets Lactobacillus crustatus.
Yes.
Any theories as to why that would be?
Like why you have displacement of CTBO5 with.
It seems that we're seeing all of these strains, these other strains, not the lactin D, but you're putting in there, and yet we're seeing more of these other strains of Lactobacillus crustatus